Okay, here we begin the process of meiosis, which is the process of making gametes, sex cells. And note, we're going to start out exactly the same way as we did in mitosis. We've got two pairs of homologous chromosomes. So here's one set. I'll flip that around so yellow's with yellow and red's with red. So same genes, two copies. Same genes, two copies. We've got this homologous chromosome pair and this homologous chromosome pair. And this would be G1. All right. Okay, so here is the cell after S phase of interphase, when all the DNA is copied. And once again, we've got paired sister chromatids. So now this chromosome is a pair of copies. This is a pair of copies. This is a pair of copies. This is a pair of copies. And they're all joined at the centromere exactly the same way they were in mitosis. Next, we'll move to prophase 1 of meiosis. So here's prophase 1 of meiosis. The nuclear envelope is disintegrating. There's actually a whole lot that goes on in prophase of meiosis. This is where crossing over occurs. Most of the movement in towards the pole of the cell happens in prophase. But you don't have to really worry about that stuff unless you take genetics after this. So we'll still stick with prophase, metaphase, etc. Now, for metaphase, the chromosomes once again move into the center of the cell. The nuclear envelope is gone. All the components to make nuclear envelope are still floating around, so you can make those envelopes later, but it's as a unit, it's disintegrated at the point. Now, in mitosis, remember, every chromosome centromere is lined up on the mitotic plate. They don't do that in meiosis. Instead, the homologous chromosomes line up at the center of the cell on the metaphase plate like this. So there's literally fingers of DNA in the homologous chromosomes, or structure, I should say, that go out and look for matching structure. And molecularly, these chromosomes will line up in dozens of different places up and down the chromosome arms, finding exact matches of structure. And that's how they find the homologous chromosome pair, so that they can line up opposite that pair on the metaphase plate. Now, the first thing you've got to go through is a division of anaphase 1, and no centromeres are split in anaphase 1. Instead, an entire homologous chromosome set, or I should say one member of the homologous pair, is pulled to opposite poles of the cell. All right? And then you go through telophase 1, and essentially this now becomes two different cells. So let's just go ahead and make two different cells here. Now look at the two cells you've got. Each cell has one copy of the blue and red chromosome and one of the yellow and red. But note that this yellow-red copy is the marked one. Over here, this is the unmarked one. Over here, this is the marked red-blue chromosome. Over here, this is the unmarked one. So these are fundamentally different cells now, all right? They have different chromosome compositions. We've now generated cells where each cell has only one of the two homologous chromosomes from each pair that the cell started out with. And so now we've got to go through a different division, a second division, so we can cut the amount of chromosomes down to 1/2 of what we started with. Remember, what we started with was this, 4 chromosomes, 2 of each type. We've got to cut that down to 1/2. Technically, these are only 2 chromosomes, but we all know that this is really 2 chromosomes joined at the centromere, so we may call it 1 chromosome, but it's meant to be 2. It has to be divided up again. So what happens is each cell goes through a prophase and a metaphase again, and during the prophase, everything's still visible, all right? And then in metaphase, you get the chromosomes lining up on the mitotic plate. We're not going through mitosis, but it's essentially the same thing. It is the equator of the cell, and now the centromeres are lining up, just like they did in mitosis. And so then what will happen is the centromeres will split, or the sister chromatids will divide at the centromere, and we'll go through anaphase 2, and we'll get the sister chromatids pulled apart as daughter chromosomes. And so let me unwrap these things. Okay, now I've taken the centromeres, essentially the joining of the sister chromatids, off. And so now we go through anaphase 2, and a copy of each daughter chromosome moves to opposite poles, pulled along on the spindle fibers in anaphase. And these are two separate cells now, all right? And they're fundamentally different. Okay, so that would be the end of anaphase 2. And then in telophase 2, you're going to start forming those nuclear envelopes around each of these new centers of chromosome density, as it were. All right, and these will eventually divide up into 4 separate cells. So let's do that. All right, I'm going to move these into 4 different plates, but remember that really what's happening is each of these 2 plates is dividing in half into 2 smaller cells that will then differentiate. So we end up with 4 different cells. And if we look at their compositions, you'll note that 2 of the cells are identical, and the other 2 cells are identical as far as what chromosomes they've got in them. They've got unmarked blue-red, marked yellow-red, unmarked blue-red, marked yellow-red, marked blue-red, unmarked yellow-red, marked blue-red, unmarked yellow-red. Real quick, let me set this up so that you can see how the other type of gamete would come from this same set of chromosomes.